So as a final speaker, I would like to welcome Dr. Lena Hausdorf, who has a PhD in molecular biology, several years of experience as a research scientist, and from 2014 to 18, she was working in the clinical laboratory for genetic diagnostics, first as the head of the deletion duplication department and later as the senior director for genotyping, where she was responsible for the entire lab process and workflow optimization. Since 2018, she's at Limbus as a clinical application manager and the head of the product management and service team. Welcome, Lena.
Thank you, and welcome to my part of the presentation today. Cabinet of Curiosities, Detecting and Interpreting Extraordinary Variants with varvis. I'll just give a very quick overview about varvis again. It's the IVD you already heard. What we do is we get the data directly from the sequencer, do the bioinformatics, and the users can then do CNV and SNV filtering based on the annotations we provide and hopefully classify and report. At the moment, several thousand samples are analyzed like this per month, and that gives us a pretty big overview about all the data that is floating around, and which enables us to actually help our customers with setting up a good workflow and also validation. For example, if I refer to the question in the beginning, we actually validate for our customers the CNV analysis, so we cannot give a general number. We are pretty good with our CNV analysis, but it depends a lot on the process in the lab, and we help our customers to set that up, and also we do the validation. And the next thing I want to say is what I will not talk about is I will not show you how the software works. If you're interested in that, please come by to our booth and we can give you a demo, or you can have a free trial, or you can go to our platform Learn Worlds, where we have nice videos where you can see, we explain how to analyze a WES, and you can clearly show and see how the software works. What I want to show you is actually more like the tricky cases we encountered and where we had some trouble. And so this is the first example I have, and I think most of you will know this screenshot from IGV. It's a whole genome sequencing sample, and looks pretty okay, but for this very, very weird spike. So we were asked, what is this? So there were areas all over the genome that were had an incredibly high coverage, and they even showed up in the software as insertions, not all of them, but some of them as de novo mutations, and nothing was detectable in CNV analysis. And when we zoom in, you see there are long stretches of mismatches on either side. So we actually wanted to figure out, is it actually an insertion? Because that was one of the theories, but a little odd with all over the place in the genome, or a contamination. And we just quickly blasted one of the reads, and it was actually bacterial contamination. And so this is something we actually haven't solved yet bioinformatics-wise, but is something that we really want to do. So this is one of our big topics for the coming year, to find a bioinformatics solution for this. And so it was a buccal swap. That's where the contamination came from. And since it's not wasn't a VGS, there was no enrichment for the human sequences, and everything got sequenced. And some of the fragments align to the human genome as well a little bit. So this is one of the topics we will address in the next year. And the next one looks a little bit similar. Again, a region with a high coverage. And we were asked, actually, this time it showed up in the CNV analysis as an insertion of 2 exons. And the problem was it wasn't confirmed via MLPA. They couldn't. They tried it several times. They couldn't confirm it. So they were like, is this a false positive result in NGS? And here, we had some arguments for an insertion, which was we have very clear breakpoints, and also there's a variant in the middle, and it had allele frequencies which matches a insertion because it was 0.3 and 0.7, not like you would normally have in a heterozygous setting with 0.5. Arguments against it were, for example, we couldn't confirm it, and it was pretty small, so they were really in doubt. And so the first hints we had were there were a lot of duplicate reads in that region, a lot. And then we asked for the primer positions of their Sanger Sanger sequencing, and they were exactly at the breakpoints. So the result was in the end. I forgot. The variant was very rare, and we were able to look in their database and see that they had sequenced it a week before. And so it was actually a contamination of the NGS library prep with a Sanger PCR product from a week before. And it's a reminder that, the worst enemy of any molecular biology is the PCR from the day before. So, that can happen in the best labs. And we were pretty lucky that there was this variant in this, because, of course, it can also happen with not just one Sanger PCR product, but, for example, 2 libraries or something. And the next one was this sample, where we had also a very weird an insertion. Here we were pretty sure, everybody was pretty sure it's an insertion, and was encompassing 2 genes, SCO1 and GRAB1L, that's what I call it. And there were extremely large insert sizes in this reads. And so we were asked by the customers, basically, are these the exact breakpoints I see here? And could it be a tandem duplication? So normally this is not a question you can answer with NGS, unless you're very lucky. And in this case, we actually were very lucky. And so what I try to, show the situation here. So you have the blue bar is the duplication or the insertion, and then we looked at the mismatched reads and blasted them again, and we saw that half of the reads was always, the upstream part was always from ESCO1, and the downstream part was always GRAB1L on both sides of the insertion. So we had this kind of situation at 2 positions in the genome. And so in the end, it basically made sense if the situation was like this. So there was a tandem duplication, and all the reads then matched at that position where the duplication was happening. And so the next case I have was, again, the duplication. They're always very tricky. And of the entire P9 arm. And the weird thing here was they did chromosome analysis before, and there was no hint of any triplication at that time. And so now we come to one of my favorite views in varvis, actually. It's one of our very extensive quality module. And I like it very much because you very easily can see here trisomies or any big abnormal thing. So you have on the y-axis, you have fractions of mapped reads, and on the x-axis, just the chromosomes. And for example, you can very easily see here on chromosome 9 that there's a duplication. It has a lot more reads on chromosome 9 than all the other samples in the run. So this is one of my favorite plots, actually. And then we also did plotted the allele frequencies. And then you can see on the left side, the blue line was a control. It clearly has peaks at 0.5 and 1, as you would expect for heterozygous variants. But the orange one was the index in this case, and it has the peaks at 0.3 and 0.7. So clearly showing that this is mosaic. And on the right side, you can see it again. It's P and Q arm. You see Q arm has the clusters of the heterozygous variants at around 0.5. And for the P arm, they diverge to 0.3 and 0.7. And the reason why they didn't find it with chromosomal analysis in the first place was they repeated it, and then they found it because the cells, it was mosaic, and the cells they used for chromosome analysis were not the same cells they used for the WES analysis. And it was only happening in a certain type of cell. And So when they redid it, they found it. The next one is a quite common question. Is we also in our QC module show you if the sex we calculate is actually matches the one that the user has given. In this case, it didn't. And so again, we have a part of my favorite plot here. And you can clearly see the green line is a normal male. It has not the same amount of, has like half the amount of reads on the X chromosome as the female. And the blue one is a female. She has less reads on the Y chromosome than the male. But the orange one was actually XXY. So it has a double amount of X that the male and also the Y chromosome reads. So that was pretty easy. And so just shows you how you can detect abnormalities on the chromosomal level with this module, actually. And something newer, that's rather a newer addition to our software, is the contamination score. It's a score that's calculated around 100,000 SNPs. It's therefore only usable for larger panels like clinical exomes, VES, VGS. Smaller ones will not work with it. And we actually validated it with the customer of us together and in vitro. So they really mixed samples, and in silico with 1,000 cases, where we tried to find a threshold to detect maternal contamination. And we were able to detect maternal contamination with a limit of detection of 1%. And the threshold you need is 0.3 for that. And actually, this is independent of ethnicity, which is pretty important. And additionally, so in this case, we had the contamination score was increased. Customer said, I don't see any contamination anywhere. I don't see it. It's not there. But in the end, they redid the analysis, and you see the other numbers you have, the normal ones that are not circled are the one of the repetition. So then the score was fine. And then we also, when we plotted the allele frequency, here because the contamination was just 12%, you're not able to see it in the heterozygous variants. But you see it in the homozygous variant on top, that they are a little lower than on the left side, which is a normal sample for comparison. We were able to estimate the contamination at 12% by using the log2 values we got for the CNV, because they actually matched the theoretically log2 value of 1.12 copy number, which doesn't exist in real life. But so this was like a contamination. We also had other cases, and that's actually very beautiful about it, where we had mosaicism detected by this. So if the contamination score is increased in our software, it doesn't necessarily mean that it's a contamination. It could also be a mosaic event of even one chromosome. We had one example where only one chromosome was actually a mosaic, and that was already visible in the contamination score. So we're pretty happy about it because it proved already to be a very valuable tool. And it's a little bit newer. And actually, I'm already done. I wanted to do this quick. And so thank you very much for your attention. And do you have questions?
So, any questions? No, not for Lena.
No, good.
There's one additional question for Roland that came in. If you maybe still want to answer this one. Is there a list of affected RefSec transcripts?
We can put that online, definitely. It's just a CSV file, it's not a problem. Maybe we can, whoever that said, maybe just contact us. We can just also send it around, but you have to realize this list has to be basically updated, with alignments and with new transcripts coming in, right? So we can give you the snapshot and the current state, but remember what Rami said, right? So also in terms of the let's say frequency that we detected, these are solved cases in our database, right? Let's say this is just the current status, right? And not maybe the future status in a year or two. Sure.
Okay. So thank you again for joining the satellite. And if you want to learn more, please visit our booth. We're ready to welcome you. Goodbye.